This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, a special episode recorded on November 1st, 2023. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Today, we are coming to you from McGill University in Montreal, Canada. And joining me here on this kind of cloudy and cold day, Angela Mingarelli.
Hello, I'm glad to be here. You're finally in my city. I've been to your city so many times now. We didn't say the weather though, Vincent.
Tell me what the weather.
So there were flurries a few minutes ago, and I think it's around 0 now. When we woke up this morning very early at 6 a.m., it was minus 2°C. I don't know what that is in Fahrenheit. What would that be?
Minus 2°C. So,
2°C.
0°C is freezing, which would be 32. So, it's about 30-ish, 29-ish, something like that.
A little bit colder than New York.
It's freezing. Yeah, it's much colder than New York.
Yeah.
And we have a great view. Mm-hmm. Hey, Karin, the regulator, do you think you could pan out the window and get a shot? How about that? We're asking you to do something out of the playbook. It's cool outside. We have a nice glass window here on the side.
Yeah, we're right next to Montreal, which is a very large park and also cemetery and mountain in the middle of Montreal, which is very
That's what the
famous
city got its name after, I guess?
Mount Royal, exactly. Montreal.
Montreal.
Yeah. Mm-hmm.
Okay, not Montreal, Montreal. But if you say Montreal in New York, they think you're putting on airs.
Yeah, they think you do.
Anyway, we are here again at McGill, as I said, and we've put together 4 guests for you, and we decided to have 2 professors and 2 of their PhD students. And so let's introduce them. On my right, Corinne Maurice. Welcome to TWiV.
Thank you. Hello, everyone.
And to the right of Corinne, her PhD student, Anshul Sinha. Welcome.
Thanks for having me.
Yeah, pleasure. All the way over next to Anshul is, we have to rearrange everyone.
Okay, sorry.
I'm going to skip over to next to Angela, Jesse Shapiro. Welcome.
Hi, welcome.
Thank
to
you.
Me and everyone.
And next to him on his left, his PhD student, Sara Naderi. Welcome.
Thank you.
Is it Naderi or Naderi? Or
Naderi.
Naderi works?
It doesn't really matter.
And Sana is correct?
Yes.
I got that right?
Yeah, that's right.
And Anshul is correct?
Great job.
All right. We're going to find out more all about you in a moment. And we have a small audience here. Thank you for coming. This is about science communication. We try to teach people how science works, why it's important for you. And if you come to my talk later today, I'll give you a little bit of the history of how I went from being a professor to a science communicator. But anyway, before we get going, I want to remind everyone that we do this because you enjoy it. And if you do like what we do, this kind of science communication, we'd love your support. You can go to microbe.tv/contribute. The company, you can see here on the video, that's the name of our company. That's a plaque assay, by the way. The greatest biological assay ever devised. Don't argue with me. Sorry. There's nothing better. There's nothing that tells you so much about a virus than a plaque assay. That's actually not a real one. It's an artist rendering, right? But it's beautiful. Michelle Banks made this logo for us. She's a great artist in the DC area. Anyway, Microbe TV is a nonprofit. In the US, it would be your donations would be federal US tax deductible. All right, and the second order of business, Amy Rosenfeld at the FDA is still looking for a second technician. The FDA, Amy is at the Center for Biologics Evaluation and Review, which is the part of the FDA where you can do research. And her laboratory works on enteroviruses, the most interesting viruses there, next to plaque assays, right? The most interesting viruses in the world. And she's interested in pathogenesis. She's made interesting discoveries about cross-reactive antibodies among the members of that family. She wants to make animal models, all BSL-2 work, and she needs a research technician to help her out there. So we'll put a link in the show notes to a description and Amy's email. You can contact her for further information. All right, back to today here at McGill University. Let's start by hearing each of your stories. We'd like to know where you're from, what kind of education you had, and how you ended up here. So, Corinne, where are you from?
I'm from Mauritius Island.
I wouldn't have guessed that.
Right? I also thought the same.
Did you know?
No, when she said it this morning, I was surprised. I always had heard your accent, but I always thought like maybe New Zealand, but then you lived somewhere else, but then I wasn't sure. So
New Zealand would have been cool, I have to admit.
Sorry, so where's Mauritius on?
Mauritius is in the Indian Ocean on the east of Madagascar. So, okay, South Africa, keep going east,
And
Madagascar,
that's the
keep
big
going
one.
east. And there's two tiny islands. One is French, it's Réunion Island, and the other one is Mauritius, which is independent. And so that's where I'm from.
You were born there?
I was born there,
This is
born and raised there, yep.
It's a tiny island, right?
Tiny.
Compared to Madagascar, right?
Oh yes, Madagascar is it's a continent, so Mauritius is tiny. I think I remember a friend mentioning that Mauritius would be the size of Rhode Island.
do they speak French like in
Which is not
Reunion?
an island.
Which is not an island, you are right, yeah.
Indeed.
They speak both English and French because similar to parts of Canada, we've had both the British and French colonies. So we started out as a French colony and then became a British colony, and in '68 became independent.
So what is Mauritius famous for, besides you, of course?
Besides me, I'd say Mauritius is famous for the dodo bird.
Ah, right.
It's the only island where the dodo, like the real one, was found. So we'd be famous for that. We'd also be famous for the most expensive postal, how do you call it, what you put on envelopes?
Stamp.
Thank you. Would be
It's going to
known.
be good today, isn't it?
I'm gonna find lose words. Yeah, because we were one of the first like British stamps that was produced outside of UK, the blue penny stamp, and that's.
And your
These are our
last
two
name-
claims to fame.
Your last name is Maurice, which is similar to Mauritius. Is there any link there? Is that just coincidence?
Damn it! I should have had by now a really nice backstory to that, but I don't. Other than my last name in French is how you would say Mauritius in French, in Maurice. It's written the same way, it's done the same way, but there were no, as far as we know, no local inhabitants in the island, so it was an imported name from somewhere.
So I presume you went to grammar school and high school
Yep.
on Mauritius, and then for college, did you leave?
College, I left, so I left the island at 18 years old and went to study in France because, not because I did have family there, but really studying in France was cheaper and I had gone through the French system. So went there. South of France, Montpellier is where I did my undergrad and grad school. I kind of stayed there. There was a little blip in the time course where in my master's I came to Montreal, not at McGill, but at UQAM, and I came to study lakes in the eastern townships.
Lakes?
Yeah, I did a year-long survey of lakes, which actually meant drilling holes in like over 15 centimeters of ice, which was a really fun experience for someone who'd never seen snow and ice for 18 years. So that was interesting. Then after my PhD in Montpellier, I went to do a postdoc in Boston for 4 years at Harvard University, and then got my position.
Who was that with?
Peter Turnbull. So that was my change in systems, because I had done my PhD in aquatic systems, interactions between bacteria and phages in aquatic systems. And then with Peter, I discovered that, well, we actually contain thousands, actually millions and billions of microorganisms. And I was fascinated by that. So I went to do my postdoc with him. And after 4 years of a very productive postdoc and really cool research experience with him, I started my own lab at McGill in 2015. I've been moving steadily north. I don't plan on going any further north, I have to admit. The weather is
Northwest
kind of
Territories is not
not
next step.
currently in the plans. I'm quite happy with the amount of snow we get here. That'll be enough for me.
It's going to get warmer and warmer up here.
That's true.
So
maybe it will be in the books,
matter.
actually,
So
in a decade or so.
What originally got you into science? Why were you interested?
Biology, more than any other science, just made sense to me. It felt logical. It felt like if I didn't see it, there was probably a reason why it wasn't happening or why it didn't exist. And I just felt as though that was just interesting. And I think all the questions I had were all in biology. I can't say I was very much interested in the rest. I think there was always a little bit of science around the house because both my parents are somewhere or another related to science. And yeah, I think it just made sense.
I agree. I can't disagree with that. It makes sense more
everyone
than
agrees
anything else. So your parents are not scientists, though, right?
Well, a physician, that is...
Yeah, not a scientist.
Not a scientist. Yeah, we were doing that.
Nope.
But we like them. We like them too.
They think they are, and the press thinks they are, but they're not.
Exactly.
And I don't want to insult physicians. But you take care of patients really well.
Exactly.
Really well.
But if you want to know about virology, do you know who you should ask?
Not a physician.
A virologist, probably, right?
Absolutely.
Not a physician. Both parents were physicians, or
No, my dad. My mom used to work in a lab. She was a biochemist, but it didn't last for long as she followed my dad traveling along. So they met in Scotland and traveled a little bit before coming back to Mauritius, because my dad is Mauritian and my mother's Scottish.
Wow, that's a great story.
That also explains more of the accent.
More of the accent there too. Yeah,
You got a little Scottish in your accent,
I
right?
do. Yeah, well, I'm half. Yeah,
Well,
but I never lived there, so
Well,
I
that's
don't
interesting.
have
I'm not
the
sure-
accent.
It's pretty far north, so.
I know.
It's true.
It's
You
probably
went
why I didn't. It's an
so
island.
your mom speaking in the accent, you picked that up, I
Yeah,
guess, right?
yeah.
Around the house, that's it. Because it's not genetic, as far as I know.
No.
To speak a certain way, you can be easily influenced. Okay, thank you for that. Let's go to Anshul. Let's get your story. You've heard now what we want to hear. So
Yeah, I don't know how to follow that up. I grew up about 30 minutes east of Toronto in lovely Pickering,
darn,
Ontario.
I was going to say Madagascar.
No, not quite Madagascar. That'd be great. We can say that though. Pickering is a kind of just suburban landscape, I guess. It's a good place to grow up. I did my undergrad in London, Ontario, not London, England, and really started to get into science then. I actually, my parents signed me up for science camp when I was a kid and I hated it. But I think by osmosis, my dad is a doctor and my mom's a science teacher, I gradually started to take a shine to biology. I took an intro microbiology course, really liked that. And then I did my honors project about 10 years ago with David Heinrichs at Western, where we studied the production of a pigment called staphyloxanthin in Staph aureus. And that was really cool to me to kind of finally have ownership over a research project. That kind of made me want to go to grad school. So I moved to Montreal, to McGill, where I did my master's with Hervé Lemoyle. We studied outer membrane vesicle production in Citrobacter rodentium. That was also a really cool project, but at the time I took one of Corinne's intro to the microbiome courses and was really kind of sold and excited by all the cool research going on in the lab. So I joined her lab as a PhD student in 2018, where I still am, studying phages in the gut.
So let me impress you. Citrobacter is the one you can use in mice as a
Exactly,
model, right?
for EHEC.
Not bad for a virologist.
Yeah.
Okay. Sana, let's hear your story.
So I'm from Iran. I was born and raised there. I was there until I was 21. I did my undergrads there. I studied engineering in my undergrads, not very biology related. But then I didn't really like it. So I started thinking about these interdisciplinary fields where you can apply computational sciences to them. And so I did electrical engineering. But then I took one term off and I went to Vienna to study population genetics. And that's when I started slowly getting into computational biology. But my background is almost entirely computational. And my project also is fully computational.
And so you're currently a PhD student here
Yeah,
in
so
Jesse's lab.
I finished my undergrads in Iran in engineering.
Right.
And then I started as a master's student in Jesse's lab originally. I actually transferred to the PhD program last week.
Oh, congratulations.
Very fresh.
Yes,
Very fresh
absolutely.
still. Yeah.
Transfer to the PhD program and you get on TWiV in the same week, right?
That's
It's
pretty
darn good.
Well, a week or two after, but yeah.
Yeah, that's good. So we don't have a good sense of what's happening in Iran, okay?
Right.
So is it hard to leave to do education like you have?
It's not really hard to leave. Most people are leaving. A lot, like from my class, we were 150 people, I think, in my year in undergrads. And I think less than 30 or 40 of them are still in Iran. So everyone tries to leave. And it's a plan that everyone has. And we start thinking about it very early on. And we start looking for programs and options. It's hard. It's getting harder in terms of visa restrictions for some students. And also our university, well, the government in general is making it harder for students to leave in subtle ways. For example, the most recent thing they started doing is that they refuse to give official transcripts in English now. So, and it's crazy because people will still leave. It just makes things harder. So it was this whole drama to get my English transcripts to McGill and satisfy the requirements. There are things like that, but generally not everyone, but most people who want to leave try to find a way to do that.
And once you leave, do you have to go back? In other words, do you have some kind of visa restrictions that make you go back?
Well, I moved here with my family, so for me it's easier in that my family live in Toronto. So I don't really go back that often anymore. Okay. For other students, they try to go back whenever they can, but it's hard. It's far. The tickets are expensive. It's not as easy it's not an easy trip. It usually we joke that door to door it takes 24 hours to go back because it's such a long flight and there are layovers and and most of us are students here, so we have classes.
And how did you find Jesse? How did you find his lab and his research, like from electrical engineering to?
Well, yeah, that's a good question. So I slowly transitioned to what I do through internships that I did. So I did a few internships in Vienna, and then I was an intern at the Genome Center in the summer of 2019 with Ryan Hernandez. I was doing mostly human genetics. But then because I knew my family's moving to Canada, I was looking for graduate programs in Canada, and I was looking in the Genome Center specifically because I used to be there. And what Jesse does is very relevant to what I was doing because it's population genetics, but for pathogens or microbes. So that's how it was. Yeah.
Cool.
So I want to ask you a question, and you don't have to answer, but how is it in Iran for women to study and advance and leave the country like you have? Is it more difficult than for men, for example?
It is for sure harder to be a woman. It's getting better. It's getting easier and it's getting better. Like in my parents' generation, it was much harder. There was women were considered less smart in general. And also, for in some families, there are restrictions that they don't really get to like go out there and experience and study and work and be fully independent. For me personally, I was lucky in that my family are super supportive and they wanted me to really like pursue what I like. in my university, it was a little bit hard because it's engineering school and it was all guys. It was 150 guys and like 10 girls. And I was often the only girl in my classes. And I did experience, there were times that they would come to me and say things that were not very nice. And you could feel that a lot of people feel like you're by default less smart just because you're a woman. And I even got direct comments on this sometimes, but it's getting better. And there are people who are talking about it and are trying to make it easier.
Okay.
But yeah.
Oh, look at you now. Here, PhD student on TWiV. We're on publications.
All right, Jesse, what's your story?
I grew up just on the other side of the mountain, a neighborhood.
We could be 1,000 miles on the other side.
Yeah.
right-
That's
right
right. And by
on.
mountain, I mean the
Hump.
hump, 300 meters, something like that.
It's small.
Yeah. So I grew up in a neighborhood called NDG, and we moved to Vancouver.
That's where you are, right?
Are you
No,
in
that's
NDG?
where my partner lives,
Okay,
not myself.
NDG, right.
I don't live there anymore, just so that I'm not exactly following in the precise footsteps of my parents. I live in a different neighborhood. But we moved to Vancouver when I was 9. But then I came back to do my undergrad at McGill when I was 17. So I felt a really strong connection with the city, and it's my center of gravity, and I always end up bouncing back here. So I did my undergrad at McGill in biology. I did my honors project in yeast genetics in Howard Bussey's lab, who's now retired, and doing high throughput mutant screens to make a genetic interaction network. And so it's this sort of high throughput science, and he was a really traditional yeast geneticist. He said, I really like this sort of like systems biology, like study the system and not individual components. And I, was interested in doing for grad school, computational biology, systems biology, this kind of thing. He said, don't do that, it's a fad, just pick a protein and study it, and this kind of thing. He was very skeptical, and I did not take that advice. I did a master's in something called integrative bioscience, which is basically biology, but it sounded fancy, at Oxford in the UK, where you do 2 kind of rotation projects, which was very fun. One was on African trypanosomes, so that was a real molecular biology project, and the other was on great tits, which are birds, which is what they call, I guess, chickadees, or some. I'm a terrible ornithologist. So anyway, that got me really interested in ecology and evolution because it was a field project. So I was out in the woods, moving birds around, and it's really hard to work with birds. It was fun, but I really got interested in ecology and evolution. And so I then went on into my PhD at MIT in a program called Computational and Systems Biology. So I got more quantitative background coming from biology. And then from there, basically never went back to the wet lab or to the field, and basically just stayed in front of a computer for 5 years, which for students out there, get a good chair, get up and take breaks. People are better at this. I threw out my back after grad school. I went on a big trip backpacking and completely threw out my back, had a slip disc, had to go back, interrupt a trip, go back and do months of physio. Luckily didn't have to have surgery or anything like that. Anyway, I should sit up straight right now. So, there are hazards of sitting at a desk. Anyway, yeah, so that's it. I did a short postdoc afterwards with Party Sabeti, and I guess because it's a virology podcast, that was the first time I'd worked on viruses. I did genomics of Lassa virus, so samples from West Africa, doing phylogenetics and evolutionary biology based on Lassa virus sequences. Also worked on tuberculosis. I work on a lot of different, I'm not a virologist, I work on lots of different things, but I mentioned the Lassa virus. And then I came back to Montreal. My first faculty position was in the biology department at Université de Montreal, which is our sort of sister French university, again, on yet another side of the mountain. I keep pointing over that way. And then I was there for 7 years, and I moved back to McGill 3 years ago in summer 2020, and in the microbiology department. I'm over in the genome center right now.
So your body may not have done well sitting in a chair, but your brain is really good, right?
Well, you know,
That's what you're using
on
all
the
the
day.
time.
hope so, yeah.
A lot of people now have these standing desks.
Yeah,
Yeah,
you
sure.
know,
Yeah, you need those.
walking, the treadmill
Yes.
desk, all of that.
Yes.
One of my lab mates, Dinesh, he has this really fancy desk that goes up and down with like a remote, and he can stand at it. It's very nice. And he has like a 40-inch screen, so everyone needs that.
Very nice.
Everyone
I don't
needs
have the
that.
walking, but I do have a
Yeah.
They're really nice.
I have a also, but there are too many wires at the studio.
right.
So it's always in a down position. Maybe we should fix that sometime so I could stand up, right? Okay, so Angela, what department are all these people in? Or what division or
What
what?
division? So microbiology and immunology,
Yep.
Dr. Maurice, so Corinne Maurice. And then you guys are in human genetics, right? Are you?
No, I mean we're all in the same department.
Oh,
We're
you
all
guys
we're
are also
all in
okay, sure,
microbiology,
sure.
immunology.
Then it's all
Okay.
But we have
Microbiology.
different. Well, we sit in different places.
Yeah.
We're a diffuse department.
Yeah.
Yeah, they're in the life sciences complex, which is where we are. Well, we're in the cancer center, but it's connected to the life sciences complex. And then the Shapiro lab is in the genome center, which is like 100 meters away, maybe 200, across a field.
Okay,
Not
and
the
what,
mountain.
what department are you in?
I'm in physiology, although my lab does mainly immunology, but our department is physiology, but we're on the same floor as the Maurice lab. And Jesse also he started on my committee, and then my project changed when I was like only
Kicked me
doing.
off.
I know, it's
I
because we were
keep
doing
it
bad
happens all the
stuff.
time. It happens all
Bad
the
stuff,
time.
and then we expanded more into T cells, and then it was like, well, maybe this isn't as relevant, but he is on other people's committees in my lab.
Okay.
And we have collaboration.
So where I'm trying to go, which we haven't reached, this center with the funny name.
Oh, the MRCCT.
The MRCCT.
Yes, the
the
McGill.
Center for
McGill's
Complex Traits.
Yes.
Are you all in the center?
The
We
The,
are.
the
Not
Maurice lab is
us.
MRCCT.
Yes.
You
We are
guys aren't
not.
MRCCT.
No.
Are you? You're on,
We're
right?
MRCCT.
So
Oh, yeah. I'm on
what
the
is-
committee of the
What is this Center
Same
for
here.
Complex Traits?
So, we were talking about this last night at dinner.
It wasn't recorded, so
It's all there, exactly. And once again, Dakota and Dinesh, people from my lab, we were saying, what is a complex trait? So the McGill Center for Complex Traits is a whole bunch of researchers. It's like 50 researchers that all form part of this center. And what is a complex trait? Well, we can say it's a trait that is influenced by more than one gene. Because at one point we were like, okay, 2 genes, but then it's not necessarily a gene. It can be, as people mentioned, an environmental factor, etc. So we'll just say more than one gene and numerous factors. So a trait that could be, so almost anything, like many things could be complex traits.
everything.
Exactly. So
Right?
it's a very broad name. Philip Groh, one of the researchers on our lab that's been there for the longest, he started the MRCCT. It was his doing, I think. Yes.
It's an interesting name. But even if you took one protein, like you were told to do, right, and just knock it, you're still going to have interactions with other things. So
Yeah,
you could say
still
that's a
complex
complex.
trait.
Environment, other proteins, and so forth.
Exactly. So, but we're the MRCCT.
Okay.
But I think that was the point of having, I mean, that's why you have so many researchers in that center with so many different expertise. So microbiology, physiology, human genetics, we have clinical researchers at the MUHC, so the hospital that's affiliated to the university. So yes, it's many different conditions, many different diseases, and I think that kind of showcases this idea of like multiple traits and multiple possible interactions. Yeah.
And it's nice because we have, I'm on the graduate student committee, and we have work in progress seminars, which, because it's so diverse and heterogeneous, all of the researchers that form part of this, it's nice to see other students work. And we actually do it in this room where we're recording, is where we have a lot of these WIP seminars. And now also the Genome Center is included in that. Now they're called Haplo Talks.
Yeah.
So
Haplotalk?
Haplo Talk, yeah.
Why?
We changed the name because now there's the Genome Center people and the MRCCT people. Before it was ATGC Talks with MRCCT, and now we're Haplo Talks.
All right, so for listeners who don't have any idea what we're talking about, right?
True.
So at academic institutions, we often have departments or divisions, which I think, and you tell me if this is wrong, the idea is to bring people together who would then interact in ways and collaborate that they wouldn't if they were just an isolated lab in a building.
Exactly.
Absolutely.
Right?
Yeah.
And so one approach to that is to have a diverse collection of people like this complex trait center, and you have some meetings and you hope that spurs interactions. Or the other approach is to say microbiology and have all microbiologists, which is gone out of favor, I think.
Yeah.
right?
Interdisciplinary is more interesting, more collaborative.
Mix
you
people
learn
up.
more.
So throw in some computational biologists,
In the
who
mix.
knows, maybe they'll be
Sprinkle them
useful.
in. Some electrical engineers sprinkled in.
Because you want innovation in the end. You want new
Yeah.
ideas, and sometimes your view of something is too myopic and someone else say, hey, have you thought of this?
That's
So that's why we have these kinds of departments. But there are other reasons too. There are financial reasons, there are organizational reasons.
Shared equipment.
Shared equipment is a good one, yeah. I mean, the departments, at least in the US, they get a chunk of money from the dean usually to run things, and so that's why it's good to have a department as opposed to individual people. So that's what we're talking about, folks, okay? Any questions? Good, I'm glad, because I wouldn't be able to answer them.
Email them. TWiV@micro.tv.
So I was in a department, when I first went to Columbia, I was in a department of microbiology, which in the 50s was made as a department of bacteriology. So that, because bacteria were the first things to be studied intensely because they were small and accessible. So bacteriology. And then they realized there's more than just bacteria, there are other microbes. And then a few years ago it became microbiology and immunology, as yours now. Is yours micro and immunology?
Yeah, micro and
because you can't take the two apart from each other. So anyway.
And speaking of that,
Yeah.
because the Maurice lab studies phages, is there anything else you wanted to say before I go into this?
No, it's good. You can take it.
What is, for all the listeners and other people in this room that might not know, what is a phage? I guess Corinne will let you answer this, and we can ask some more other questions to Anshul after, but what is a phage?
Well, it's a sort of virus.
Okay.
It's as simple as that. Basically, a phage is a virus that is only going to be able to infect bacteria. It can be recognized by our immune system or by eukaryotic cells, but it usually won't infect them. So it doesn't have the machinery, it doesn't have the way to recognize and infect these cells, whereas bacteria, they can. So we study the viruses that don't make you sick, or that
Or maybe.
indirectly can make you sick.
I was going to say, isn't that, this, we'll talk about it, but there's this IBD linked to certain phages.
Yeah, exactly. So as I was mentioning earlier, what interests me in studying phages is really their interactions with bacteria. Like I've always been interested in how these two, kind of like predator and prey, tend to interact with each other. And I used to do it in aquatic systems, but at the end of the day, you find these microorganisms everywhere. And in the human body, they can have really strong health or impacts on our health. And in some cases, you would want these phages to stick around because they would be in charge of like killing your bad bacteria, for example, the ones that can make you sick. So these would be the phages that you'd want to keep. But because these phages can also be recognized by your immune system, or some of them would actually kill your good bacteria, those are the ones that could potentially make you sick, but it would be their indirect effect. And we've had several projects with different conditions, including IBD, and that was a project that was started a while back, but that Anshul took the lead on. And so I'll defer to him to actually talk about that because he did incredible work.
I want to just highlight the paper itself. Anshul has his first author on a paper from 2022 in Microbiome titled Transplantation of Bacteriophages from Ulcerative Colitis Patient Shifts the Gut Bacteriome and Exacerbates the Severity of DSS Colitis. So, bacteriophage is synonymous with phage for all the listeners.
Yep.
And explain what this means. What was an overview of this publication and your research?
Before you do that, I have a very basic question. So all of us have bacteriophages in us.
Yes.
Right? Free-living, free-floating bacteriophages as well as integrated into the bacteria.
Absolutely, we have the two.
So on my skin, I would have some phages too?
Yep, everywhere.
Everywhere.
How many?
About similar to how much bacteria we have.
Which is how many?
10 to the 13 per gram of
feces.
stool.
That's
so
our reference point.
20
is
pounds of
per stool.
Yeah.
We have a pound of bacteria. How many pounds is it? I heard once this crazy number of like microorganisms in our body was Do you
And
guys,
I do have it on my slide to the first year undergrads, and I right now cannot remember like the weight of it.
but is it over a pound?
It's, I would say, approximately
Like
a pound of
chunk of butter?
microorganisms.
Yeah.
Yeah.
And it's about 1-to-1 bacterial cell per human cell, and then
human
you
cell.
count 1 phage per bacteria.
It'll
Yeah, so if you bring it to the human body, for each like human cell, you have 1 bacterial cell. And on average, for each bacterial cell, you're going to have 1 phage. However, if you get out of the human body and like you look at like the Earth in general, for each bacterial cell that you're going to found on Earth, there's going to be approximately 10 phages. They can be the same or they can be different phages. So it's not only abundance or their numbers, but it's also their diversity and the types of phages that are there. So there's something very interesting about the human body. Like, why isn't this 10-to-1 ratio there in the human body? That may be due to the immune system. It may be due to the whole like system itself. But yes, the phages, bacteriophages and phages are, for the moment, the most abundant
entity.
entity. There you go.
It's crazy to think
In
that
us.
we are just
In
as
us.
much
And on
phage
Earth.
as much as we are human.
Yep.
That's
So-
like self.
So are these in our blood as well, these phages? Or
I
they
guess
must-
that's a bit controversial. I think there have been some studies that have sequenced small amounts of phages, but mostly they remain gut.
How about my brain? Do I have phages in my brain?
Shouldn't.
You
That's a
probably do.
Yeah,
I don't know about the rest.
Well, I have on my brain, yeah, for sure.
probably not.
Okay.
What about other barrier surfaces? Like you said, the skin, yes, but in the lungs there must be tons of phages, right? Like a whole other lung virome
Phagome.
of phagome, I guess, in the lungs.
It's not really been studied too in depth. I think the gut's been the first logical place to look for phages. We're still kind of in the early stages of phage discovery in the human body. It's
Okay.
a field that's only 10 to 15 years old. So I'm not sure about.
So the papers that would be looking at phages in the lung have typically done so in, for example, cystic fibrosis. So you have the disease that's there, and you're trying to understand, like, is it because of some microorganisms that are there, overgrowth of some of them? So typically there, yes, we're finding phages, not very abundant, not very diversified, but it could be, this is a challenging body location to sample. And contamination is a big deal there. So yes, we have usually started with organs or locations where you can easily access the community. So gut through the stool samples, the skin, because it's really widely accessible, oral, so the mouth. There have been a limited number of studies, but they still exist for everything that's urogenital tract. But they're also, like, accessing the samples isn't always easy for different reasons, I'm going to say. And then in the mouth, what's interesting too is that you've got like the actual mouth versus the teeth, that there does seem to be quite a strong difference there. And then lung is one of the low ones. And I'm not aware inside the body. I don't think phages, well, they haven't been shown to do it yet. I'm open to the idea that we're going to find them, but we haven't seen them yet.
My colleague Elio Schechter on TWiV used to always say, you know, it's the fecal microbiome, not necessarily the mucosal microbiome, right? It
Absolutely.
could be different. It's hard
Yeah.
to sample that, but feces are easy to sample. That's why we do
Absolutely.
it, right?
Yeah, yeah.
So the reason I'm asking all this is, if you take serum from people, do you find antibodies to any of these resident phages?
I'm going to defer the immunology questions that way because this is, as I keep saying to students, I'm not an immunologist.
There have been phage therapy papers that have looked at, you know, you give someone a dose of
Yeah.
phages and then you look at antibody titers and you do see, at least transiently, the production of antibodies.
And this
that
is
are
an
pretty-
oral dose
That's
of
true.
phages?
Yeah.
Yeah.
Like a drink of phage?
A drink of
phage
phage.
drink, phage slurry, we can call
Well,
it.
also they give it IV, depending on
Also,
the,
could do-
Yeah,
yeah.
there's a
You can
depending
do
on the
IV
disease.
therapy
Yeah.
with phages.
But I'm just curious if you make antibodies to your resident phages, or since you grew up with them, I assume you get them at birth and shortly after. Maybe they're viewed as self, right? That's the question.
That's the question, and there are several teams trying to look into that because we're starting to get a good idea of where we get the bacteria from at birth and how we get these bacteria, how we acquire them, what makes some of these bacteria stay in the gut versus some of them just like they're lost of the system. And now these same questions are coming with the phages, yeah. Okay. And for the moment, we are assuming that we're getting them at birth through the bacteria. So you were talking earlier about free phages and the ones that are integrated within their bacterial host. That's a way for phages to replicate. They basically can multiply in different ways, either directly infecting and killing or integrating and staying kind of like protected by the bacterial cell. And we think that's what's happening at birth, that we first get the bacteria. Once they're settling in the gut, plenty of signals, which we don't know what they are, but these signals, environmental, maybe body, maybe immune linked, unclear, stress the bacteria, cause the production of these new phages who start establishing in the system. And then for anyone who's seen an infant, it's very clear that the rest is going to be environmental and like direct contact with the parents, the environment, the floor, whatever goes in their mouth, really. But yeah, that's a super interesting point.
All right, so now back to you.
And
Back to your
IBD
In
work.
the
research.
paper, this paper, how did this come about, as Angela said?
Right. So I guess it's been known for a couple decades now that the bacterial communities in the gut in inflammatory bowel diseases are very important. The changes that we see in patients in certain types of bacteria are really important in causing excess inflammation. And what's been found in the last sort of 5 or 10 years is that the phage communities are also sort of altered in these patients. But at the time, we didn't really know what the consequence of those changes were. So we wanted to see how the phage communities from IBD patients were interacting with bacterial communities and how that might be different from phage communities from people who don't have IBD.
So IBD, inflammatory
Bowel
disease. So
disease.
what kinds of diseases are included there?
Crohn's disease and ulcerative colitis are the two sort of main diseases under the umbrella of IBDs. So in this study, we were specifically looking at ulcerative colitis patients. So what we did is we took germ-free mice, so sterile mice. We colonized them with fecal bacteria. And then we gave those mice phages either from ulcerative colitis patients or from sort of non-IBD controls.
Are these human? So you took it from, human.
Yeah, we took human
ulcerative
stool
colitis.
samples, colonized these in mice. So the mice were kind of our vessel to do these experiments.
These are people without IBD.
So the bacterial communities were from patients who had IBD. And then we took the phages from either IBD or non-IBD. And then we did sequencing to look at how the bacterial and phage communities were changing over time in the mice. And then we also, in collaboration with an immunologist at McGill, Ira King, we looked at the immune responses in the mice too. So basically what we saw was that the sort of healthy and IBD phage communities were shifting the bacterial communities in different ways. For the UC phage communities, we saw increases in certain sort of pro-inflammatory bacteria like E. coli, and a depletion of certain bacterial taxa, like we saw Eubacterium decrease, which is sort of protective in the context of inflammation. And sort of in addition to those changes in the bacterial community, the mice that received the dose of phages from the UC patients also had higher disease severity. So we gave them a compound called dextran sodium sulfate, which induces colitis in mice. And we saw that the mice that received those sort of UC phages had higher severity, which suggests that these phage communities might be sort of playing a role in disease progression, possibly through altering the bacterial community to a somewhat pro-inflammatory state.
So how do you prepare these phage preparations from people? What do you do exactly?
Right, so we get our fecal sample, we take it to our anaerobic chamber, here 2 floors below, and then we separate the phage and the bacterial communities using centrifugation. So we pellet the bacteria, and then we're left with a phage supername that we filter, which should contain sort of the majority of the phages in the fecal slurry.
And do you look at the distribution of that phage population at all to see what it is, for example?
Yeah, so we sequenced the phage inoculum. So phage like understanding what's in a phage community is a little bit of a challenge because there's so much diversity worldwide of phages, but we can get some sort of signals of what the phage community looks like. And it was pretty consistent with what's been seen in other studies in terms of the IBD phage community. So we see a lot of temperate phages that can undergo both the lysogenic cycle, meaning integrating into the bacterial chromosome, and the sort of canonical lytic replication. So that's a hallmark of the phage community in IBD patients, and we saw that in our own sort of fecal slurries as well.
So when you take phages from UC patients and put them into mice and then treat them with this chemical,
Right.
it the phages from the UC patients exacerbate the disease caused by that.
Exactly. Yeah. So
What is it, DSS?
it's DSS colitis. So it's not a perfect model.
What do you say?
Clinicians won't say that it's a great model
No.
to study IBD, but it's useful for our approach where we want to sort of induce colitis. We want to see how
Right.
the phages are changing the bacteria and then induce the colitis and see how that affects the mice.
So when you say it exacerbates, like what are we talking about? Forget about statistics. Just, is it
Right.
really good?
The mice don't want it. They lose a lot of weight. So the mice that got those phages lost more weight. And we also saw pro-inflammatory cytokine production in those mice increase.
Now, and in the mice receiving phages from healthy people, you don't see this.
Yeah, well, they do exhibit disease symptoms, but at a much lower level.
And the pro-inflammatory response is lower as well.
Exactly, yeah.
And there's a shift in the microbiome as a consequence of
As a
this, correct?
consequence. And we see during inflammation compared to before inflammation when they have the phages and bacteria,
Yeah.
during inflammation, those changes are even greater. So in the context of an inflamed gut, it seems like these phage bacteria interactions are really sort of expedited.
Did you characterize again the bacteriome after the challenge?
Yeah, we did
With
it
the
at kind of at all stages, like baseline, after we add phages, but before the chemical induction, and then also after. The
And you
chemical.
saw an increase in pathogenic bacteria?
Yeah, sort of
communities?
throughout, and then it kind of went up sharply with the onset of colitis in certain cases.
So maybe you were asked this when you had the paper reviewed, but how do you know there's something else, there isn't something else in the phage preparation that's mediating this effect?
It's a good question. We did a heat-killed control as well, so we heat-killed these phages. So what we're left with is kind of just the supernamed of everything except the phages, and that was similar to our PBS controls,
All right,
I
that's
guess.
good.
Okay.
That's a
Yeah,
good
so that was a good sanity
Although
check
the
for
heat
us.
killing could kill a toxin.
It could kill other things, so it's not perfect. We did check endotoxin levels.
I'm reviewer number 3.
Yeah, I know. I thought we were over.
It is
It's been
now.
published for 2 years almost. Vincent.
Sorry. So if you let's say you give mice phages from UC patients,
Right.
what if you followed with phages from healthy patients? Would that reverse the sensitivity to DSS?
It's a great follow-up study that I'd love to do.
Yeah.
Because, yeah, I think the idea in some people's minds is that phages from healthy donors can be used as sort of a therapy because they might, like Corinne was saying, kind of infect and reduce the abundance of some of those more pro-inflammatory bacteria in that context. So I think that'd be really cool.
I guess you could also give microbiome fecal transplants as well. And you
Exactly.
would, along with that, you would give the lysogenic phages, and that would accomplish the same thing, right?
Right.
And it might be better.
Right.
But people have done these kinds of studies, right?
Yeah, it's been done. I mean, in C. diff, for example, there's been fecal virome transplants that have been shown to be quite effective. I think the problem with IBDs is that fecal microbiota transplants in and of themselves are not that effective.
Okay.
So it's hard to imagine that for patients, giving them a dose of phages from a healthy donor would really cure everything. It has to be, I think, a multifaceted approach with something targeting the immune system and then maybe a microbiota-based therapy kind of supplementing that.
So you sorry, go ahead.
You had a really good point earlier about our immune system actually recognizing or not the phages. So basically our immune system recognizes or should recognize our phages as like self. And in IBD and all these other chronic diseases where you do have an immunology defect or an immunological issue or a challenge, your immune system is not responding the way it should. And so you're not having the microorganisms that should be there, nor the phages. So phage therapy, or just proceeding with an FMT in that case, will not be the magic solution because your immune response is not going also in the direction that it should be. So it's definitely more challenging in the context of these diseases. But an infectious disease where you have a fully functional normal immune system, every FMT that has been tried seems to be working much more effectively and much better. But really the poster child for FMT, so fecal microbiota transplant, or even going the step further of FVT, fecal viral transplant, is Clostridium difficile infections. Like those are the ones where it works really well. That's the ones where we're trying, well, other teams are trying to develop therapies. And then it's just a case of, can this be applied to other infectious diseases? And I think we should try.
So this has been done, or is it ongoing now, fecal virome transplants? So you called it FVTs.
Yeah.
And how is it, I guess there's a challenge of the stomach, then is there some sort of are these encapsulated in pill form? Like practically, how does this happen if it is happening in humans? If this is ongoing?
All of the above. Okay. We're still at the stage really where we're testing things out to try and identify the best way to approach this problem if ever we were to give it out as a therapy. So right now, FMTs, so the whole microbiota transplant, are done, but it's really the last resort treatment. So you'd have to consider a variety of parameters, but it is done. The FVT, so this time just focusing on the viral part of things, I don't know to what extent it's actually done, other than the research papers that show the really good success and the promise of it. Then there are several labs that are looking either trying to find like the perfect donor, so the human volunteer, where this time you would transfer like the whole community. Other labs and companies are actually considering making synthetic communities. So this time it's lab-based complex bacterial and viral communities. So you know what's going in, and that can be provided as a pill form, can be provided in some specific foods in some cases, but then it goes like, which bacteria should you be adding in? And then like only bacteria, or which viruses, or should yeast also be part of this? Like, so it gets complexified, and other teams are just like totally going the other way, where instead of giving you microorganisms, it's like, okay, can we actually modify what's already there? And maybe trying to use phages this time as a delivery agent. Like, we know that phages are going to reach that stage. Can we just give the medicine or the compound with these phages? So all of those are actively being pursued by research labs, by companies. In our lab, we're trying to just, I guess, put some of the pieces of the puzzle together, or just adding more puzzle pieces. We're really early on trying to understand, because at the start of Anshul's project, we didn't know if these phages were even going to have an effect. It could be that they wouldn't do anything because it's all about the bacteria. His work showed that no, phages are actually actively playing a role. Not the main one, because it seems to be dependent on the bacteria, but without them, the symptoms are less strong. So they're clearly driving something there. But that's in mice. How does it translate to humans is right now an open question that we're going to be looking into maybe one day.
And apart from IBD, because you said that your lab also has other models, other disease models that are possibly indirectly or directly affected by phage populations, and which are those? Apart from IBD, you said was which includes Crohn's disease and ulcerative colitis. Are there any other models that you work with?
Right now, we've taken a shift towards early life development and childhood stunting. So childhood stunting is a condition that actually It's a worldwide problem, a man-made problem. It's linked to malnutrition. And really 1/3 of children under the age of 5 worldwide experience malnutrition. And malnutrition in the sense of not getting enough food or not getting good quality food. So there's a variety of reasons why that's happening. But really one of the consequences of child malnutrition is stunted growth. So children who don't grow to their full potential. And it's not only physical, but it's also mental and cognition development that's taking place. And it is a condition that is multi-generational. Child who is stunted and develops into a stunted adult will most likely have stunted children too. And the issue with that is that, well, stunting is just one of the conditions of malnutrition. You have more severe forms of malnutrition, but basically this condition puts you at higher risk of developing chronic diseases. It puts you at higher risk of catching infectious diseases and so forth. And once again, we have been showing that phages are an important member of your gut microbial community in the sense that if you don't get the right phages during early childhood, you are setting yourself off in a not wrong trajectory, but not being able to fully develop the way you should be. And that is, you're not going to have the correct immune responses. Once again, you're not going to meet the correct developmental milestones at several levels. And we currently have a project looking into this early life system. And for me, in terms of like microbial interactions, it's fascinating because we're looking at early in life. So the whole system is setting up. Like microbially speaking, the bacteria, the immune system, the phages, the viruses, they're all kind of like fighting with each other and recognizing each other and kind of setting the stage for what's going to be happening earlier on.
So importantly, maybe this would be breastfeeding versus bottle feeding and cesarean birth versus vaginal. For the listeners and people just in general,
All
that's
of
very
those.
important in
Yes.
in colonizing your gut, and then when it's permeable when
Absolutely.
you're born. And this
Yeah.
is what you're referring to in your
Absolutely, yeah.
And your diet can influence this as
And
well.
your diet
right?
can influence this
So if
as
you
well,
have a
yeah.
poor diet, as you were saying, leading to stunting, that could influence the phages. So potentially phages could help fix some of that. is the idea.
Potentially, yes.
Do you have a
that's-
mouse model for that?
We have just developed a mouse model for that. So that's the work of another graduate student in the lab, because the current mouse models of malnutrition typically use adult mice, which is great if you want to know what's happening with mature communities or adults. But this whole aspect of if you're a stunted adult, you're going to give birth to what is a high chance of being a stunted child and so forth. And this whole like 0 to 3 year window, that's what was missing with our animal models. And so right now we have actually developed, we're still waiting on the sequencing data, but we do have, once again, germ-free mice, so the same mice that Anshul has been using. So they've never seen microorganisms in their life. So these are not animals that you would find outside. They don't exist outside of a lab. And they allow us to ask us these cool questions of like, what happens when you give some specific microorganisms to these animals? And so in our case, we've been giving infant stool samples to these adult mice. So we know that there's not a match there, but we have these adult mice with these infant microbial communities. We breed the mice, so we let them reproduce, and then we're actually following the pups from birth to weaning. So when they actually stop breastfeeding from their dams, from their mothers, and that's where we're looking what's happening. How are the bacteria establishing? How are the phage establishing? Does it change if we give a different diet to the mothers? Because obviously these pups don't eat the solid food yet, and so forth. And so we're just starting there, but it's super exciting, and there's lots to come there. I'm excited about it.
Very cool work.
So Anshul, we're listening here about maybe one day having phage therapy, right?
Right.
But how much is your phage different from my phage, right? Is it going to be the same for everyone, or is it going to have to be tailored to a certain extent?
I think it'll have to be tailored to a certain extent. Between like in the room, for example, there's not a phage probably that is shared between all of us. There's a high level of inter-individuality in phages. With that said, a lot of phages between different people can infect potentially the same bacterial host. But I think if we're thinking in a disease like IBD, I think understanding what's wrong in the bacterial community, so maybe if I had IBD and I had a lot of Klebsiella, which is known to cause higher levels of inflammation, then maybe we develop a phage cocktail that's specifically targeting Klebsiella. So I think that's kind of the direction where things are going. There's a few clinical trials going on now with phages targeting certain bacterial sort of pathobionts in IBDs. But yeah, I think it's going to be more of a sort of personalized approach.
Also, isn't it part of it knowing what the bacteria are contributing in terms of metabolites and so forth? And maybe that's the end goal that you want to modify, right?
Right. Yeah, exactly. So I think that is a bit more difficult, and we have ways in the lab of kind of understanding what the bacterial are doing at a metabolic level and activity level. But I think the first thing would be to sort of target the ones that we kind of know are causing disease.
So what are you doing now?
I am trying to wrap up my PhD.
You'll get there.
I'll get there, yeah. A couple months to go. And then, I guess the world is my oyster. We'll see what the next steps are. But wrapping up my PhD, and sort of the other part of my project is understanding the switch of replication cycles in phages during inflammation. So I talked a little bit about sort of what happens with the phages after the community has been altered. But how do they get there in the first place? So we think that prophage induction is possibly driving this switch. So when lysogenic phages are integrated as a prophage into the bacterium, prophage induction is the process by which they excise and replicate. And that happens in the context of bacterial stress. And during inflammation, there's a lot of stressors in the gut. There's increased oxygen levels in the gut. Bacteria are exposed to a number of metabolites that they wouldn't otherwise be exposed to. So we think that stress might be driving prophage induction and sort of causing a shift in the community.
What do you want to do next in your career? Do you know?
That is a fantastic question that I'm doing soul searching as we speak to try to find that out. So I love science. I love working on this project. So I want to continue to work ideally in the field of the microbiome. I think it's a really cool system to study. My project, I touch on bacteriology, virology, immunology, computational biology. So it's a really cool system to really try different things. So I really like that aspect of it. So ideally stay kind of in the microbiome field.
But you want to do postdoc next? Is that
Yeah,
the goal?
that's probably on the horizon. So
Okay.
yeah.
All right. Angela, should we move on to
Yeah, let's
Jesse
move on.
and Sana?
Yes, so Jesse and Sana have, Sana is first author of a publication in eLife that was published in April of 2023, titled Zooanthroponic Transmission of SARS-CoV-2 and Host-Specific Viral Mutations Revealed by Genome-Wide Phylogenetic Analysis. So, what does this mean? Because for the listeners out there, there's a lot of big words in this title and we need to dissect these. So Sana, can you please explain a little bit about your research?
Very simply put, what we were looking at was how frequently is SARS-CoV-2 being transmitted between humans and animals. So that's the transmission analysis part, the first part of the title, that's what's referring to. Another analysis that we did was that we then looked at genomes of SARS-CoV-2 that were taken from animals, and then we were trying to find mutations that are specifically adapted to different animal hosts.
Okay, so which animals did you guys do analysis
So
of,
there are
or
a lot of
genomes?
animals that have been infected by SARS-CoV-2. I think a year into the pandemic, I don't remember the exact number, but there was a report saying more than 50 non-human mammals are able to be infected by SARS-CoV-2. But then at the time when I started this project, I looked through the data sets that were available, and the only species that we had enough data of were cat, dog, minks, and deer. So white-tailed deer, farmed mink, and cats and dogs. That's the 4 species that we looked at.
Okay.
So you didn't do any sampling yourself, right?
No,
You
no.
got sequences from databases, correct?
Yeah, so there are public databases of SARS-CoV-2 genomes, and they were being updated regularly, and it was a large data set already. We didn't do any sequencing ourselves or sampling ourselves.
Would you like to? Okay, that's
It's
enough.
okay to say no.
I think I will say no.
We had a lab retreat in the woods
Yeah.
last weekend. We saw a couple deer, and so
Yeah.
I thought, well,
Yeah.
Define had a swab.
yeah.
That's what we were.
You can't catch a deer.
Jesse
No
and I were
way.
saying.
You run fast.
Yeah.
They'll run away from you.
We also
yeah.
thought stay away from it. Yeah, you know what?
Probably has COVID.
Yeah.
Probably has
No, but
COVID.
this is a good point because there's so much data available that you can mine it, right? And that's-
Right,
that's
exactly.
what you did.
Yeah. Yeah, I think when I was doing my project, there were around 1,500 animal host sequences, and there were around 8 million human-derived SARS-CoV-2 sequences
Yeah,
on the
yeah,
public database available, which is a very large number.
So before we talk about this data, so Jesse, you did other things before SARS-CoV-2, right?
Yes.
And so why did you decide to do this, and what did you think you would contribute?
Yeah, great question. I swore after my work on Lassa virus, I said, that's it, I'm never going to work on viruses again. I'm going back
Here we
to
are.
bacteria, and I work on many different bacterial species and some pretty basic questions in ecology and evolution using microbes. I'm never going to do this again. But the pandemic started, and I just said, around April 2020, surely someone is sequencing the viruses in Quebec and building trees and things like that. And I happened to know someone at the Quebec Public Health Lab, Sandrine Morera, who I'd met at a conference the year before. I said, hey, are you guys doing this? And she was like, we were doing it. Yeah, sure. Do you want to help? We're trying to do it. And I was like, all right. So I sort of got involved a little bit with the provincial effort that then was coordinated into a more federal effort across Canada to sequence and build phylogenetic trees and just sort of do the surveillance. So yes, despite my best effort, I sort of did get involved.
Because you have skills in computational analysis, right? That's the point.
Yeah.
Right.
So we're sort of at the end of this kind of workflow, right? Where people were getting swabbed, things were getting sequenced. It was going through this whole workflow, and then we were kind of analyzing the data, building these phylogenies.
So I have 2 questions for the listeners that I think are very important considering the title. What is a phylogeny, and what is zooanthropognosis?
They could each answer one.
Exactly. You can each answer, you guys can choose. But I'm sure there are many people wondering, what do these things mean? Phylogenetic analysis, what does that mean?
I can do phylogeny.
Sure.
So phylogeny means, a phylogeny is basically a tree, a family tree, that describes the genealogical relationship between things. And you can make phylogenies of different species, or you can make phylogenies of different individuals within the same species, or you can make phylogenies of the same gene across different individuals. So it's basically a tree.
So you use sequences
To build phylogenies.
But in the old days, before sequences, we could measure parts of animals and use that, for example, to make a phylogeny, right? Basically. Yeah.
But we used genomes, whole genomes of SARS-CoV-2.
And you have programs that have been written that you use for that, right?
Yeah. So there are so many pipelines that construct them based on what you're inputting into them. The one we used is a maximum likelihood algorithm that just tells you what is the most likely tree given this data that you're giving it.
So if you have 100 sequences,
Yep.
the program will do what? It will put the closest ones next to each other?
Pretty much.
Is that it?
Yeah. So the ones that are closer will end up closer to one another on the tree. The way it's done, it can be the algorithm or the process through which the tree is generated can be different. But yeah.
And that you also try and infer an ancestor, right?
Yeah, yeah.
Even though you might not have the ancestor, you say this is the ancestor of these and so forth.
So in our case, we were lucky that we knew the ancestor, the Wuhan strain that was sequenced in December 2019, that we knew that that's the root of the tree or the ancestor. But yeah.
Okay, so that's phylogeny.
Yes.
Yeah.
And then if I can, well, so I think this is linked as well. And so Sana's using these phylogenetic methods and using existing methods. But one thing that she did was actually to write some computer code to essentially parse this information, right? And then on the tree, you have information about is it a human or is it, let's say, a mink, right? So we'll do sort of one animal at a time, right? So you have so each leaf on the tree is a virus that was sampled either from a human or from a deer. And then there's a method called ancestral state reconstruction, which is basically going to walk down the tree and say what was the ancestor given these leaves of the tree. Was the ancestor more likely, was it in a deer or was it in a human if you trace back? So Sana wrote some code to basically trace that back and label those ancestors, those internal nodes, saying, was it probably in a deer or probably in a human, to be able to then say, what's the most likely scenario of, did it then go from a human to a deer, and then from a deer to a human, tracing along the tree, where the tree sort of actually measures these transmission events that we didn't observe, but we're trying to infer how those transmissions happened along this tree.
Yeah, because when you have a phylogeny, you only have the data or the information for the tips or the leaves of the tree, but the internal nodes, we don't have them. They're in a way, you can say they're ancestors of those individuals that we didn't sample. So we don't know if they were taken from an animal or from a human. We will have to estimate that based on the information that we have from the tips of the tree and the topology of the tree. You have to estimate what is the most likely state for each of the internal nodes. And then you have to browse the tree to look for transitions from one state to another. And that's when an actual transmission from a human to an animal or from an animal to human probably has happened.
So, for example, if you have a human sequence and you find this is unlikely, but just to illustrate, you find the exact same thing in a deer, you can infer that it came from a human, right? And then in the deer, you don't agree with it, that's okay. And then in the deer, then you have some changes that are never found in humans. So you would assume that those are deer changes. And then if you find any of those in people, it suggests that virus went back into people. Is that how it works?
So it's a bit more general than that. So we took whatever deer sequence we could find, and then I tried to match those deer sequences. When I say deer sequences, I mean SARS-CoV-2 sequences that were taken from deer. And then I matched those with all the human SARS-CoV-2 genomes that existed at the time to find the closest relatives or the sequences that are most closely or most similar to the deer ones. And then I built a tree of those sequences pulled together. And then when we have the phylogeny, then the sequences that are closer, genetically closer, tend to cluster closer to each other on the tree as well. And then when we do the ancestral state reconstruction, and then we look at we identify the transitions from one state to another, that's where we infer that a transmission has happened. We don't explicitly look at mutations directly to infer transmission, but that's implicitly included in the phylogeny, because when sequences are genetically more similar, then they cluster together more closely, and they are more likely to those are the branches along which the transmission has happened.
So if you have several lineages here, tips, and they're all human, then there's a deer one in there.
Yep.
That means they had a common ancestor, most likely.
Yes.
Right?
Yeah, so we have to estimate, is that common ancestor more likely to be a human ancestor or an animal ancestor, based on the topology of the tree and the information we have from the tips. And then wherever an animal node has a human child node, that's like tree terminology, but like on the tree, their nodes have children, or those are branches that come out of them, right? So if an animal node has a human child node or tip, then we would guess that's probably a transmission from an animal to a human.
All right. So you had about 100 deer SARS-CoV-2 sequences?
Yeah,
So you download all of
150.
those, right?
Yeah.
And then how do you find which human sequence? You find the closest human sequences to those for your
Yeah.
analysis? Is that it?
So let's say we have 150 deer sequences. I created a distance matrix between those 150 deer sequences and all of the 8 million human
So
sequences.
you did 8 million.
Yeah, that's like that's a matrix that's 150 by 8 million.
Wow.
It's a very big matrix.
Does that take a long time to?
Yeah, I think it took me a week to calculate that
So
matrix.
the computer is just computing.
So yeah, I was running it.
Yeah, you're not sitting there for a week at your computer.
It's
broken
good.
down.
I didn't do it. But, like I didn't do it manually. I ran it on Compute Canada clusters.
Okay.
The
So very
HPC
powerful
cluster.
computers, right?
Yeah, but even on those, it took multiple days because this is not something you can parallelize a lot. So because you will have to go through every single
I see.
sequence,
You
and
can't do this on your iPhone.
no, not even on my laptop.
No, years ago, I asked Michael Rossman, a crystallographer, he said, how do we used to not even have the computing power. What do you do now? And he pulled his iPhone out. We can do a lot of the structural analysis on my iPhone.
I wish I could run that on my iPhone. My life would have been
You can log in to the cluster.
I can log in to your
analysis.
An app. Yeah.
that
you got a
actually
terminal app.
So because you need to block a chunk of time on the cluster for multiple days, usually you might stay in the queue for a long time. And I remember during that week when I was submitting these jobs, I would wake up because like in the middle of the night, like no one submits jobs. So sometimes I would like set an alarm and wake up at like
submit a job.
2 a.m.
2 a.m.
to submit jobs, and then I would check if it's been like queued. And sometimes when you submit a job, there might be like a small typo or a tiny error that you don't load a module, and then it will fail. So you wait 5 hours in the queue, and then it won't run, and that's so annoying.
You know, it hasn't changed. When I was in college, we used punch cards
Yeah.
to submit jobs. And if you had an error, you'd come in and get your printout, and it failed, and you'd have to. It's the same thing. Nothing's
Yeah.
So while this is happening, you're doing other things. You're not sleeping the whole time, right?
Well, I
night?
was not doing much during those 5 days because that was like the bottleneck that I had to that was the main thing I was waiting for. But yeah, usually if I'm running something that takes long, I try to keep myself busy in other ways.
And what were some of the interesting findings? 'Cause we haven't gotten there yet. So what are some of
The results for so of the 4 species that we looked at, we found that transmission rates are really high in the mink to human direction. So mink tend to infect humans very frequently, whereas
That's because they're farmed, right?
exactly because they're farmed and they interact with workers on the farm. Whereas for deer, we inferred very low transmission from deer to humans, and that's because they're wild animals and probably they don't get to come in contact with humans that often.
Unless they're killed, right?
Yeah.
But human to deer, like we did a paper recently on TWiV, there's so many deer now, almost all the white-tailed deer that they've collected samples from are infected with SARS-CoV-2 or have.
Yeah.
So there's definitely one-way
Yeah,
transmission happening.
Yeah, so in the human to animal direction, we found that the transmission rates are somewhat similar and they're kind of high. We have a scatter plot in our so the whole point of this is comparing different species. So we found that in the human to animal direction, it's fairly consistent. And we found around 50 events, more or less. It's like centered around 50 or inferred counts in the human to animal direction. But in the animal to human direction, the transmission is very variable. So minks have high transmission rates, deer have low transmission rates. For cats and dogs, it's trickier because almost all the data that's available for cats and dogs are pets. So pets that got sick or their owners got sick and they had them sequenced. So the data is very sparse and random in a way.
How many cats and dogs were sequenced? Like in the, what was the end of, was it like 5 cats? Was it 500
No,
cats?
it was
dogs?
around 80 cats and 50, 60, less than 100 cats and dogs.
Okay.
We originally downloaded more, but we had to do quality, like quality filtering of our data, and a lot of the sequences are just randomly deposited onto GSAID, which is where we got our data from, and some of them very low quality. I remember I had to throw out so many cat sequences because the genomes were not complete. And so, yeah.
So when you say the transmission from deer to humans is low, but it's not zero.
It's not zero. We inferred it, but so the median, I think, because we had different replicate runs to count the transmissions, I think we found between 0 and 1 or 1.5 was the median, more or less. Very low, but not zero.
So does that prove it, do you think, or just imply it?
I know of several studies that have reported with high confidence that deer transmitted COVID to humans. I think there's the Ontario deer paper that with a high level of certainty.
It was all based on phylogenies, right?
Exactly. Yeah, I don't think there's any real, traditional epidemiology linking, right? So it's true that it's all inferred, but I think it's, even if it's rare, right? I think there's lots and lots of deer, as you said. So it's rare, but probably not zero, right? So it is something to think about as this virus goes and evolves off in different directions in different animals, including deer, that there's a non-zero chance that it'll then come back into humans. And this is, I guess, sort of motivating this whole study. Why do we care about this? Right? Is that as soon as a virus infects a different animal, it can go off on a whole new evolutionary trajectory that may or may not have anything to do with how it will infect humans, but it will sort of start sampling a new set of mutations, a new evolutionary space that can have unexpected effects when it comes back into humans.
So that's why we need to continue surveillance in non-human animals, right?
Exactly. And I think, Sana said that, there is, since our study, there has been a lot more deer sequencing, for example, which is great, other animals, but, at the time there was something like 8 million human sequences or viruses from humans. Now it's over 15 million, I think. So we're doing pretty good at sampling humans, which is great, and that should continue. Maybe not at the same super high level we had during the peak of the pandemic, but I think that there's a lot of potentially very interesting stuff happening in animals, and those are under-sampled. So we could stand to sample more.
So Sana, as more data comes available, do you look at it and add it to your analysis?
So we're not looking at animal sequences of SARS-CoV-2 anymore.
Okay.
The project's, I think, pretty much finalized. We did think, so we are doing some other analyses of within host dynamics of SARS-CoV-2 in animals, and that's almost finalized. We did think about adding the new deer data because there's a significant amount of deer data available now that we could add. We're not sure yet, maybe.
So one of the things you show in the paper, you have single nucleotide variant SNVs that associate with deer, right? So presumably these are changes that are fixed in deer. How do you know what they mean, and how do you know if there are any consequence for people? Is there any way to look at that?
I think the clinical consequences of these mutations in humans, we don't know of, unless there's experimental follow-up of these mutations. I don't think we can make a judgment unless we could guess based on where on the genome they are and what protein they affect. We could maybe make a guess, but I don't think without experimental follow-up we could.
You could do experiments in cells and culture and see if they make any. I mean, that's not great, but it's all you can't really infect people. That's the problem, right?
Yeah, that's true.
But in deer, you could, right? People do infect deer experimentally. So if you had some SNVs that you thought might be important, you could test that. Not you, but in general.
In deer.
Yeah, in deer.
Right.
Yeah.
You would need a very expensive setup to have that.
Well, some people do. There have been
True,
challenges,
yes.
and that's how we know that in
They
deer
challenge
infection
things.
is relatively mild, right? Because they do challenge in deer, and so, yeah, it's very expensive and it's hard to do, but it can be done. So if you had a single nucleotide change that you found in all deer, it caused an amino acid change, you might want to say, make the virus and see what it does. It would be interesting, right?
Yep.
Because there can be neutral changes as well, right?
Yep. Yeah, for sure.
Did you know, by the way, and I love to mention this every time this comes up, the cat story, when they first found out in the UK that the virus was going into cats, Angela knows where I'm going.
Yes.
Boris Johnson wanted to kill all the cats in the UK.
Of course he did.
Of course he did, right? Of course he did. Wow.
Can you
Thankfully,
imagine?
he did not.
Can you imagine they come to your house and take your cats? Many people love their cats.
I mean, they did it with mink farms,
Yeah,
right?
they did. Yes.
So,
Yeah.
There's no more mink farming in the Netherlands
Yeah.
now.
Yeah. They
that
called
was-
all the mink.
Yep, there was a huge industry.
Yeah.
So,
Yep.
and, I mean, I guess it's because it's a non-essential industry, right?
Well, it's also not domestic. It's not someone's property. It's not someone's animal. Because even though, like, our pets are considered our property,
Right.
right? This is an industrial farming. This is
But viruses come from pigs, but we're not thinking of not doing pig farm because we need it for protein,
oh no,
right?
of course.
So that's the difference between minks.
Mink is just for clothing
And
and,
pigs.
for
Anyway.
But interestingly, you guys found, talking about these SNVs, these SNVs, there was 26 in deer, multiple in minks, but you didn't find any in cats or dogs. Maybe, Jesse, can you comment on this? What do you think is happening there in these domestic species?
I think so. Mainly, I think the main thing is a caveat, which is it's a smaller sample size, right? So again, with more sampling, it doesn't mean they don't exist. We could find them. So it could be that we're just statistically underpowered. So I think that's the first thing to say. It's a strong possibility. Other possibility, if you want to make some interpretation of the data, is that these are mostly dead end infections, and so there's not a lot of time for adaptation. There's not multiple cycles of cat to cat to cat to cat transmission, which is sort of how we interpret the deer results, which is there seem to be a lot of cycles of deer to deer to
yeah,
deer
they
transmission.
live in herds.
A lot of transmission, a lot of time, lots of transmission cycles in deer, so there's sort of time to adapt, and that might be less the case in a cat where a person infects their own house cat, but then
That just stops there.
that's the
There's
end.
no other cats
Yeah.
for it to then continue.
Yeah.
the
So that's
transmission.
that's one interpretation.
We've only sampled small numbers of non-human mammals. Do you think lots of other mammals are infected out there that we don't know about? You ever thought about that?
Squirrels of Montreal.
Probably. I do
Raccoons.
think. Yeah. No. I think
Squirrels.
this is a very broadly a generalist virus, right?
Yeah.
It doesn't care that much. It's maybe a little that makes deer less sick. Like that there's some variation,
Yeah.
viral loads. But broadly, this is a virus that kind of thinks, oh, mammal, all right, no, this is great. You've got some kind of an ACE2 receptor.
I got
Right?
this.
Yeah.
I mean, I think rodents are most likely widely,
Yeah.
and they're not so hard to sample, right?
Yeah.
It would be really interesting. I think we should look,
Yeah.
right?
Has there been any sampling in rats? I think there
Rats?
was in New York, right? In rats,
There's
they did
So-
check.
And I think in New York, the sewage had some cryptic lineages that they thought might be rodent, because we have so
Right.
many rats in New York.
Yep.
So now when I
Yep.
see rats on the subway, hey, do you have some SARS-CoV-2?
And the rat's like, so
And
do
other
you.
things.
That's right. Exactly right. But I think it's way more widespread than we know, but we should find out. And there's not a lot of sampling
Yeah.
initiatives going on. People don't seem to care until something happens. And then, in retrospect, in hindsight, whatever it is. But
2020.
I think we need to do that. So, Sana, what are you doing now? Anything you can talk about?
Yeah. So right now we are doing wastewater stuff, very exciting wastewater things. So I'm working on a project with two of the postdocs in our lab so there has been extensive sequencing of wastewater in Quebec. So we have
SARS-CoV-2, yeah.
SARS-CoV-2 wastewater sequences over the course of, I think, a year almost, like 370 days in different cities. And so what we're trying to do is to see if we can forecast changes in case counts using the information that we can get from wastewater sequences of SARS-CoV-2.
Is this from 2022, the data you have? 2021?
Yes,
Or when is...
I think it starts mid 2021
Okay.
until late 2022. For some cities, I do have sequences as recent as early 2023, but I think the consensus is around 370 days starting from 2021.
Okay.
So we're using the sequences that were taken from sewages in Quebec, and then the case counts that we have for these cities, and doing some time series, time series stuff, and see if we can find a way to predict or improve existing predictions.
Do you see any of these cryptic lineages when you do this?
We're not. So we did do some exploratory analyses of the lineages that exist in these sequences, but we're not really focusing on lineages that much right now.
Okay.
We're mostly looking at the SNPs and the diversity
Got it.
that we find in the samples. Yeah.
Yeah, wastewater is a great predictor of.
Yep.
There's this website, I forgot the name of it, that Daniel Griffin always refers to. We look at through the U.S., you can see it's divided into 4 regions, and the Northeast has just kept going up and up and up, and finally, and he blamed it on the Italians,
He's blaming us.
He said Columbus Day, all the Italians got together. But I think it's a really interesting predictor of what's going on, right?
Yeah.
So you're a relatively new PhD student, but I'll still ask you, what do you want to do next? Have you thought about it?
Well, first I'll finish my PhD.
Yeah, that would be good.
Please,
sure.
please. I encourage you to do that.
yeah.
It's only been a week. A week or two. She's just starting. I mean, in the PhD program. Officially.
Well, technically now I'm a second year PhD
yeah, you start
student.
in PhD too, right?
Yeah. Oh, well, as of January, I'll be a third year PhD student. Yeah. We'll see.
But you have skills now that can be applied to many fields. You have
That's true.
computational skills. They don't have
That's
to be viruses or bacteria. They could be anything. Human genomes, right?
I like viruses and
Good.
bacteria.
That's good.
Right answer.
Nice.
I like viruses too.
I really like. No, I do like what I do right now very much, and I would like to continue doing the same thing. And what I always we were actually talking about this with Jesse this week in our meeting. What I was saying is that there's so many computational tools that have been developed for other fields, such as finance or econometrics or neuroscience or engineering, and all these very powerful tools that can be applied to so many things, so many problems that we look at in Virology.
Yeah, it's true. If you Google
Genomics.
Time series forecasting, it's
Right.
all
Yeah.
stock. It's all stock market,
all
right?
Yeah.
That's
Yeah.
that's all people want to do.
And no one's using them in the genomics or biology world
Why
as
not?
much.
Why not?
And I think it's because I don't. People are scared of the math and the
Agreed.
yeah.
In my case, I agree. A lot of us don't really like math.
Don't
Yeah.
you want to
Leave
win
it
the
to
stock market?
My
Yeah.
father's a mathematician. He can do that, but it's true. I think it can be intimidating as well. Like a lot.
Yeah,
of the,
and I often see this reluctance to go for really complicated, not complicated, but really math heavy or computationally heavy methods, but they're very useful. And I really like going to the other side and finding something that's useful on this side and then
That's
bringing
good.
it over.
Great. Well, I went into biology because 40 years ago there was no math.
That's not true. My dad's a
not true.
mathematician. We're the
There's
same
a
age.
I don't know.
There's differential equations.
There's no math in the
40 years.
biology I did. And now it's different. But I don't do any research anymore. So, you're in academics, obviously. Do you think you'd like to stay in academics or maybe do industry?
I don't know.
Not yet. It's the same. You could ask you the same question. Right? You don't know.
Yeah, I think there's obviously pros and cons to both. I think being a grad student, you're a little bit underpaid over the course of
A little bit.
a little bit.
Yeah.
Nicely put.
Go ahead,
So,
you can definitely say it.
is TWiV a safe space?
TWiV is safe. TWiV is safe. I am also a graduate student.
Yeah, so I think there's pros and cons. I love being in academia, just being surrounded by other academics and so many cool research questions all over the place. So pros and cons, and personally, I'm still trying to figure that out myself.
What about you, Sana?
I think I would say I'm a little bit impatient, and I prefer more immediate results to like long-term results, maybe. That's why I like the nature of industry work maybe better, because I do something and I see the results as it contributes to something more quickly, whereas in academia you have to like maybe wait years or decades.
It's the long game. Definitely.
I might change my mind in 2 years, but
No, we're going to hold you to it. This is recorded.
Written in
Online
stone.
forever.
You can't change. You can't change.
Yeah.
Anshul, you have any more questions?
Yeah, well, I wanted to ask both. Well, we have a question for Corinne and Jesse after, but I'll ask Anshul and Sana first. What advice, let's say Anshul first, what advice would you have for future graduate students, for undergrads, high schoolers, even anyone that's looking to go into graduate school? Do you have any advice or insights?
I think one thing is just, number one, make sure you're getting into a situation where you're surrounded by good people, because I think the environment in grad school sometimes isn't thought about enough. Obviously, you're going to do it because you like the science, but I think also just being in an environment where you are supported and are supported for the things that you do and appreciated. I think that's really important as a grad student.
Agreed. I think that's a very good answer. Your supervisor, the students that are in the lab, meeting them before starting in a lab multiple times. I know a lot of people, at least in my lab, I met with 4 different people at least 3 times before I even agreed or signed anything. So
yeah, you're there for 5 years, which
at
is
least,
kind of insane.
At least, yeah.
You signed something?
No, I signed something. I mean, I don't know. It's true.
You said yes.
You said
I
yes.
could leave any day.
It's a verbal agreement.
It is a verbal
Something to
agreement.
get your big paycheck,
Exactly,
right?
application. You have to sign the stipend form.
Yeah.
It's a little
Yeah.
sad.
Yeah.
That's true. Yes, we do need their approval to
Like,
give them the
agree to-
little money they get.
But yes, I think that's really good advice. Yeah.
But Sana, what's your advice?
I think my main advice would be to explore and don't be afraid of trying out different things to find what you like. Because as someone who did this huge shift from electrical engineering and system dynamics to viral genomics, I can say that there's so many different things out there that you can try, and right now the world is a crazy place because everything is so interdisciplinary, and you can do everything in so many different ways. And I feel like, so for me, it was my undergrad experience was difficult in that I didn't like what I was doing. So in the process of trying to find something that matches my interests, but also I'm skilled in, was a journey. But I was often intimidated by I'm in the Faculty of Medicine in the Department of Microbiology and Immunology. I haven't had a single biology course since 7th grade. But I learned through osmosis, right? So I think my main advice is that don't be afraid to explore and find something that you like, because there's so many things out there to try.
So don't be intimidated, right?
Yeah,
That's
yeah.
good. All right.
Exploring is fun.
All right, that brings us to our last questions. Angela, you want to start?
Yeah? So, Corinne, what would you have done if you weren't a scientist? What would you have been? Something else?
Most probably. What exactly? I don't quite know. I need to think about that one there.
Have you ever thought about a parallel life? Or Jesse, if you have something, you already maybe have a whole story in your head. You're like, I dream about this every day.
Yes, yeah, exactly. I think this, or can I jump in while you think?
If you already have it,
Yeah. I think this is a bit of a cliche for scientists, but I'll go with it anyway, which is to open a coffee shop.
Oh, yeah.
Yeah.
We love coffee.
And I think it's sort of a grass is always greener. I love coffee, right? I like, well, I'm not that good at cooking, but I like preparing stuff, and I like making coffee, and I like coffee, and I like working in coffee shops too. I like, or like just a different, I like coffee shops.
Would this be like a $9 latte coffee
Definitely.
shop? Or would it be
This is just like pure profit.
It's not like a $2 filter
exactly.
coffee. This is nice, like oat milk lattes with like all.
Exactly.
Okay.
What's with the oat milk?
I like oat milk.
So at least make Americanos, because the coffee shop we went to, they said, no Americanos.
It's an Italian coffee shop,
Oh,
and they're
yeah.
very strict
They
about
won't make
being.
Americanos.
No.
About being,
Only like short
Yeah.
espresso.
Yes, exactly.
They wouldn't even
Okay.
put ice in your coffee. They think
No,
that's
no.
Respect.
terrible.
Real purist.
Yeah.
Terrible, yeah.
Very purist
Real pure,
Italians.
yeah.
They're also testaluda. They're hard-headed Italians. So I can say that.
I learned in Italy that also if you order a cappuccino in the afternoon, it's like, are you crazy?
Yes.
It's not breakfast. Like cappuccino is for breakfast.
Exactly.
In the afternoon you have a little espresso, but like, what are you're crazy.
Yes.
Order
Doing that.
Wow.
Okay,
But
so
this
barista.
is the first coffee shop answer in all the years we've been asking this.
Okay.
That's
Okay. So maybe it's not
Yeah.
maybe
It's
it's
I've
not
heard
basic.
someone say that though before as well. But not.
I've
I
heard
don't
other.
remember
I've
who.
definitely
Cool.
heard it before.
All
I
right.
could see you maybe in like 30 years. We'll find you in like
Sure.
Little Italy somewhere
Yeah.
with a coffee shop.
Yeah.
Okay.
Have you come up with something?
Well, I think mostly there's the cooking part that I like doing, so I would have probably ended up in a restaurant somewhere. But also I love explaining things, and I can just ramble for hours. So probably I would have ended up as an educator.
Podcaster.
Or podcaster.
Podcasters.
There you go. Not sure I like the sound of my own voice, but definitely talking is not a problem, and trying to make a point also. So yes, I probably would have been a teacher. I'm not sure I would have had the patience with for young children. I'm learning that I am actually more patient than I thought, but I think yes, education somewhere to always be able to just talk all the time. I think that's.
Well, you spoke very well today on TWiV. So I'm sure.
So you like talking.
I do.
All right, I have a proposal for you. I'll get back to you.
Okay. But I have a
We'll find
question.
something for you to
I
do.
have a question for Jessie, because you've had both the Lassa virus and now SARS-CoV-2. I want to know, would you go back to study more viruses, or that's it? Did
Remember who's
SARS-CoV-2?
Yeah.
Remember who's listening.
I don't know. There's I think that there is, and to be dead serious. No, but there is, and to Sana's point as well, sort of how broad and a fun part of research and being an academic is. You don't really have a boss. If you get funding to do something, you have a club. You do it. No one. So you can work on as many different things. No one's going to tell you not to. So I love that you can go, you can try different things, right? But so for me anyway, there's always this tension of sort of spreading yourself too thin.
Oh yeah.
And then there is a nice tension of sort of not being the expert. You're always learning something, right?
Always.
So I'm not a virologist. Talking to virologists, who I should say on Sana's paper, her co-advisor Selena Sagan is a proper virologist, right? So having these conversations, you're learning so much, right, about a new system. So I love that. But then there's also you don't want to be too much of a dilettante. I'm working this. You don't have enough time, right?
Yeah.
So that's not a firm answer. So would I work on another virus? Yes, under the right circumstances. But you don't want to do
Don't
too
worry.
much.
We'll
We have
get you
another
into
pandemic.
Phages.
Oh yeah.
We'll come
Yeah.
back to
Yeah.
you.
Exactly.
So, Jessie.
Yeah.
It sounds like you would have a good collaboration.
Oh,
right?
we would. We just haven't managed to. We've been spitballing ideas for several years now, but nothing actually happens. As you rightly said, you do need the funding at one point to kick in and to support some of these cool ideas that we have. But yeah, we're just waiting. There's going to be a moment, I'm sure.
That's right.
All right, it's time for us to wrap up. This is a special episode. By the way, when I say special, of course it's special that we travel and have guests, but I don't know what number it's going to be, so I'm hedging. I don't give it a number. Special episode at McGill University. The show notes you can find at microbe.tv/contribute. If you want to send questions or comments, you can send them to TWiV at microbe.tv. And if you enjoy our work, once again, please support us, microbe.tv/contribute. It doesn't have to be a $9 latte or whatever it is. It could be $1, which would be less than a cup of coffee anywhere, right? You can't
It's
get
very hard to find, even at Tim Hortons. I think
give us
$2 at
$1.
least.
Give us $1 a month. Microbe.tv/contribute. my co-host today here at McGill University, Angela Mingarelli. Thank you so much.
It was wonderful being here, especially with all of these wonderful researchers at McGill. And we'll see you on Friday. We have another TWiV.
Yes, we record every Friday at 3 p.m. New York time, and Angela comes on once a month, right? Mm-hmm. So she'll be on this time. I always look forward to it. I love having these conversations. Our guest today from McGill University, Corinne Maurice, thank you so much.
Thank you for having
joining
me.
us. Jesse Shapiro, thank you so much.
Thank you.
Anshul Sinha, thank you.
Thank you.
And Sara Naderi, thank you so much.
Thank you.
And good luck to both of you in the rest of your careers here and beyond.
And good luck, Jesse, with the coffee shop. Potential teaching position.
Right.
Teacher
Good
there.
luck.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.